This is our machine learning group project, identifying plant species from herbarium specimens. There are 3 members in our group, including Wang Chenmiao, who is from Department of Physics, HKUST, Ming Xue, who is from Department of Chemistry, HKUST, and Jia Zhan, who is from Department of ECE, HK PolyU. You can see our codes on GitHub. In this folder, it contains the Python scripts and the IPython notebooks we use to deal with model. In this folder, you can see the paper details. And then I will start this presentation. The table of contents contains the background of the tasks and the difficulties we come across in this problem. And Minnie will give us an introduction on the methods and in this task and the results we get. Then I will make a conclusion. The background of this task is that we want to practice and better understanding about different architectures in convolutional neural networks. As we all know, convolutional neural networks is widely used in industry and academic research because it can deal with a lot of tasks like image classification, action identification. It works quite good to deal with things about image. We want to find an open data source for academic research, so the story is that we find an ongoing image classification competition on Kaggle, which is Herbarium 2020 FGV-C7. We need to identify plant species from herbarium specimens. The date contains a lot of images with COCO format descriptions, and the images are all botanic specimens. 1 million of them are labeled with a lot of information. We treat them as training set, and 130,000 are unlabeled. We treat them test set. The size of the image dataset is quite huge, which means it may need a lot of computational resources. The descriptions of this dataset contains annotation, category, image, and region information and license. The target of this competition is that we need to predict specific category for a given specimen image. The task is quite difficult. The first thing is that we need a lot of computation resources to deal with such a big number of images. However, we are lack of GPU clusters, so we can only use the online resources. For example, the Kaggle notebook and the Colab. They provide some GPU hours online. The second thing is that it is a multi-class classification, which means it's not a simple task. For example, you tell the image whether it's a cat or a dog. It's quite a difficult task to do a multi-class classification, especially for 3,000 and 32,000 categories. And the dataset, what's more, the dataset is biased, which is a typical problem in biology. It differs a lot from the usual machine learning tasks we come across. In this picture, you know that most of the categories only contains around 30 images. However, the number of each category varies a lot, from only 1 picture to more than 1,000 pictures. So we introduce some methods in this task we will use. For example, a multi-step model versus a single-step model. When we deal with the problem, use the whole dataset or the subset of the image that will be introduced by me.
Before we started to train any models, we need to pre-process the dataset. Firstly, we took a look at the raw images from the original herbarium dataset. We found them at different sites, and they also contain the black margin, which do not have much information that can help us to predict the final categories. So we decided to remove the black margins and also uniform these images. So we crop a square shape at the center of each image and also resize these squares to the smaller ones. We used the batching processing API from Keras. This API can help us to process the dataset quickly. Actually, we also considered using some data augmentation tools to help us augment the dataset, but also considering our limited computational resources and the dataset has already been large enough, so we do not contain that part in our practical training. Secondly, we need to integrate all the description and information from the COCO format JSON file. We got a pandas data frame for all the specimens. There are 3 labels and 1 image for each specimen, and the 3 labels are at different resolutional classification. Family is the most coarse classifying standard, and category is the finest. Since we don't have any labels for images in the test set, so to evaluate the performance of our models, we further split the training dataset into training and validation parts. The split ratio is 90:10. We used convolutional neural networks and their variants such as ResNet to help us train the herbarium dataset. There are several reasons why we use CNN. The first reason is because of their excellent performance at extracting image features. They can construct complex features from the previous primitive features. The second reason is because CNN take advantage of the spatial correlation between images. They can detect fundamental elements like edges and corners. So we can easily use a model someone else has trained as a pre-model in our own training. Later, we will show a case that we use ResNet 50 and the parameters tuned on ImageNet in our own training. The third reason is because they use filters to share parameters, so the computation complexity has been reduced. As we said before, this project is quite challenging because of the large input dataset and also because of the multi-class classification problem. So we decided to start from a toy model. We chose 100 images from the first 22 categories to form a mini dataset. We built a shallow CNN model for it. We also add dropouts to avoid overfitting, because this dataset is small. But different from the conventional CNN models, our model used a 3-step approach to predict the final category ID. We borrowed this idea from a notebook on Kaggle. The model also trained to predict for family and genus. And the fine classification training is only triggered when the prediction for coarse classification reached a certain accuracy. Here are the training results. We can see from the plots that the training is pretty good. For the training set, we get low loss and high accuracy. And for the validation set, the loss is still quite low, but the accuracy curve seems interesting. We still get a very high accuracy when predicting family and genus. But when predicting category on the validation set, though the accuracy for training set is high enough, but the accuracy remains quite low, almost only 10% for the validation. It seems an overfitting problem happens. After a test at the toy model, we began to train the whole dataset. We used the ResNet 50 and the pre-tuned parameters from ImageNet. We fixed those parameters during our training. It reduced the computation complexity. And we add additional output layers to the ResNet 50. In the first model, we decided to predict a category directly. So there's only one output layer, and the output vector has 32,000 dimensions. The hyperparameters are shown on the right below corner. But according to the training curve on the left, we can see that though the training accuracy keeps going on, the validation accuracy remains almost zero. So it is not satisfying to use this model to predict the category ID directly. So we trained a second model on the whole dataset. We used the same three-step approach as the one we used in the naive CNN model. We set the triggering condition for next classification training at when the current prediction accuracy reaches 5% and 2% respectively. The left is the training plot. The plot shows that on training set, the family accuracy can reach about 11%. The genus accuracy can reach around 5%. But the validation accuracy is not as high as the training accuracy. For category prediction, the accuracy on training and validation are very worse. Especially on the validation set, the accuracy remains zero all the time.
The conclusion of this project is that, first of all, we find the weakness of deep learning when we deal with a problem, for example, this task. Deep learning is not a good solution when we handle the categories with small samples. The second thing is that the model is overfitting if the validation accuracy cannot be further optimized. The third thing is that, combined with the research background, the biology can tell us something. Family here is a more general standard. However, the category is too fine-grained. The separation of training dataset and validation dataset is very tricky, especially when we deal with some category with small sample size. There are several ways we can adopt to improve our model. First thing is that the dataset should be divided into different parts by the sample size of each category and drop out some categories with small sample size. The second thing is that different machine learning model can be trained on each part to avoid the weakness of deep neural networks on biased datasets. The last thing is that combine several models trained on small datasets, then do the prediction will help us make the model more robust. This page shows the references for this project. That's all. Thank you.